one of my favorite chapters, which is the nervous system. And we're going to pretty much spend the remaining portion of the semester kind of focusing on different aspects of the nervous system. All right, once we'll do muscles, but then we kind of swing back into the nervous system and talk about several different functions of the nervous system. And then we finish up with chapter 16, which is special senses, sight, sound, taste, hearing, all that. So we get to learn about what's going on. Okay, so let's start off talking about basic overview of the nervous system. Back in chapter 1, we discussed, all right, homeostasis. And the homeostasis is based on, all right, the general functionality of the nervous system. We start off with the receptor that's going to transmit information to, oh, I'm sorry, guys. I didn't even have the thing on. You guys could have told me. All right, I apologize. Let me turn this on. There we go. Okay. So going back to chapter 1, you have your receptor that's going to monitor a stimulus. It's going to take that stimulus information and then transmit that input to the control center. And then that control center is going to process that information and figure out how it's going to respond to that information from the stimulus. And then it's going to send its response information as output from the central nervous system, the control center, down to an effector. Okay? So we're going to kind of follow that here. Right? So I told you that 2 of our control center systems are going to be the endocrine system, which is going to use chemical information, and then the nervous system, which is going to use like an electrical information. Okay? So we're going to talk about some of the actual functions here. So keep in mind, all right, the nervous system uses that electrical activity in the form of what we call an action potential, okay, nerve signal, okay? And it's going to be one of your major communication systems throughout your body, all right? But it definitely plays a large role as that control system there. So it's going to get that information, process it, and then figure out what its response is going to do, okay? So when we start off, all right, with the nervous system, a significant portion of its main functions is to collect information, okay? That information is whatever the stimulus is, okay? Stimulus could be something that's hot. So you touch something that's hot, okay? I know it's hot and it's painful, okay? So the stimulus that you are going to experience can be outside of your body, in the external environment here in this room, let's say, or inside your body, okay? For example, if you eat some food and you keep eating the food and you keep eating the food, your stomach starts to fill up and fill up and fill up, and it stretches. And you have these specialized stretch receptors in the wall of your stomach to tell your brain, I'm not hungry anymore. So you get that full sensation, okay? So that would be an example of a stimulus in the internal environment of your body, okay? But all in all, that information that gets collected is going to wind up in the spinal cord and the brain. And what is that? That is your central nervous system. Okay? When we break down our nervous system, we break it down structurally. That's the anatomical form of it. We break it down into the central nervous system, which is made up of the brain and spinal cord, and then the peripheral nervous system, which is everything else. Easy to remember. Just know that the central nervous system is the brain and the spinal cord, and you'll be good to go with what the peripheral nervous system is, okay? So once we collect that information, okay, we're going to process it and figure out what our response is going to be, all right? We're going to determine how we will respond to that sensory input, because that is that information that we've collected. We refer to that as sensory input, okay? And then the central nervous system is going to initiate a response. So one of the 2 structures, all right, the brain and or the spinal cord, are going to send what we call motor output, all right, from the central nervous system to the effector organs. And for our purpose, the effectors are going to be either muscles or glands. That includes all 3 muscle types, all right, skeletal muscle, smooth muscle, cardiac muscle, okay, and all the glands. All right, so I'm trying to keep it simple because we're going to get into much more detail here. All right, so collect the information, process and evaluate that information, then figure out how we're going to respond to that information that we collected. Or how are we going to respond to that stimulus? All right? So when we talk about the central nervous system, like I said, we're going to break it down into the central nervous system and the peripheral nervous system. Those are the two parts. You can see it here on this picture. Okay? So in our central nervous system, we got the brain and the spinal cord. And then our peripheral nervous system, we have nerves. And when I say nerves, named nerves, all right, are going to be part of the nerves, like the sciatic nerve, okay, the femoral nerve, like the musculocutaneous nerve. All these named nerves. So if you ever hear somebody say, oh, my ulnar nerve got entrapped, blah, 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 that's part of the peripheral nervous system, okay? And then ganglia, all right? If you remember, and if you don't, that's okay, I'll repeat myself, from lab class, all right, when we were talking about the nerves, all right, a nerve is a parallel bundle, okay, of neurons, all right, and a neuron is a nerve cell, okay, and that's going to be in the peripheral nervous system. Well, when we have a collection of the cell bodies or a clustering of the cell bodies, that's going to be a ganglia. Okay? So you need to know the definitions of those. Nerves, parallel bundle of axons in the peripheral nervous system. The ganglia is going to be a cluster or a grouping of cell bodies. And I'll tell you what's going on in those clusters there. So as we kind of zoom in on this and take a close peek, here we can see the structural organization. Remember what I said, when you see structural, think of anatomy. Okay, so you got your central nervous system, brain, spinal cord. All right, then the peripheral nervous system is going to be all these nerves, all right, that we see coming off of the spinal cord and the nerves coming off of the brain. Okay, and then the ganglia, where the cell bodies of some nerves are located. All right, now let's talk about the functional organization, and that is the physiology, how things work. Okay, so we break it down into two components, sensory and motor. Now remember, the sensory organization is all the incoming information from the external and internal environment. That's going to be that information going, all right, to the central nervous system. The motor is going to be all that output from the central nervous system, all that information leaving the brain and the spinal cord, going out to the effector organs, muscles, and glands. So when we talk about the sensory nervous system, all right, because we're going to break down, I'm going to come right back to the slide. When we break it down, all right, we have the sensory nervous system, that's the input information, and then we got the motor nervous system, that's the output information, okay? So those are our two areas, all right? And then we further subdivide those areas down into somatic sensory, visceral sensory. We're going to get into that. But I just wanted to show you what the heck I was talking about. All right, so back to this slide. All right, so the first division of the functional portion of our nervous system is the sensory. All right, also known, you want to know this, afferent nervous system, okay? So the receptors that are located both for external stimuli and internal stimuli, all right, will be stimulated, and they're going to transmit information to the central nervous system. All right, so when we break down our sensory nervous system, all right, into our two further subdivisions, we've got the somatic and the visceral. Now remember when I told you way back in chapter 1, when you hear visceral or viscera, think of organs, okay? Stomach, liver, spleen, pancreas, bone, all right, muscles. Those are going to be, all right, your viscera, okay? When you hear or read somatic, think of the body, but more so think of what you can consciously or voluntarily control. So the somatic sensory nervous system is going to detect stimuli that you're consciously aware of, okay? If your chair is hard, if it's soft, all right, if your clothes are scratchy or itchy, if it's hot or cold in this room, all right? Also, vision, okay? These are that you can consciously perceive. So that will be a part of the somatic sensory system. The visceral sensory system, okay, are going to be things that you do not perceive. You do not perceive, all right, when your heart rate goes from 55 beats a minute to 60 beats a minute, okay? You do not perceive when some of your blood vessels vasodilate and vasoconstrict slightly, okay? So the visceral sensory is going to detect stimuli that you do not typically or that you are not consciously aware of, okay? So that's going to include heart rate, all right, BVs or blood vessels, they're vasodilating and constricting, your kidney output, all right, if you're producing, all right, if you're excreting more sodium in your urine compared to more calcium, you will not notice that, okay? All right, the other division of the functional organization for your nervous system is going to be motor, okay? We also refer to it as the efferent. You're going to get some sort of effect from that, okay? And so this division is the output, specifically the motor output from the central nervous system to the effector organs, muscles and glands, okay? So when we're dealing with the somatic motor, remember, somatic, it's something that you can consciously perceive or voluntarily control. Okay, the only thing that you can voluntarily control motor-wise is the skeletal muscles. Okay, you're doing it right now as you're writing. Okay, you're doing it right now as you're cracking your knuckles. All right, if you cross your legs, whatever. That is going to be the skeletal muscle. All right, and that's part of the somatic motor system. Okay, autonomic. Okay, it's like autopilot. This is going to be your visceral motor. All right, so you will. This is going to pertain to anything that is involuntary. Okay, so what's involuntary? Smooth muscle. You can't control smooth muscle contractions. All right, heart muscle. You can't control your heart rate. All right, you start to exercise. Try keeping your heart rate low. You can't do it. All right, and then the glands, endocrine glands, exocrine glands, okay? So that's going to pertain to these tissues, heart, smooth muscle, and glands. Now later on, in chapter 15, we'll talk about the divisions of the autonomic nervous system, called the sympathetic and the parasympathetic divisions. Sympathetic is the fight or flight, parasympathetic is the rest and digest, all right? These divisions will get into much more detail, all right, in the later chapter. All right, so here's a picture, all right, of the functional organization, all right? So you got the sensory nervous, motor nervous, somatic sensory, that's conscious perception, all right? And then you've got visceral sensory, that's not consciously perceived. Then you've got somatic motor, this is voluntarily controlled. And then autonomic motor is not consciously, all right, controlled or involuntarily controlled, okay? All right, so let's ask you a question here. What are the two primary functional divisions of the nervous system? All right, we just talked about it. Sensory and motor, okay? How do they differ? Sensory is going to bring, all right, receptor information that detects stimuli, all right, both internal and external, to the central nervous system. Okay, so it goes from the receptors to the central nervous system. The motor is going to take the information, the output, all right, the motor output information from the central nervous system to the effectors. Okay, it's important. From the central nervous system, this is to the central nervous system. Does everyone get that? Pretty good. Am I going too slow? I can speed it up. All right, just want to make sure. Okay. This next part, we already talked about a little bit, so I will kind of speed it up here because we talked about it in the lab. All right, just a quick review, all right? The nerve, again, is going to be a bundle of those axons in the peripheral nervous system, okay? We talked about this in lab on Tuesday, that there's coverings to different parts of this nerve, okay? I'm going to start in reverse order, and this time I'm going to start down here in the endoneurium. I'm going to start small and go big, okay? So you've got an individual axon, which means you have an individual neuron. Right? A neuron is a nerve cell. Okay? So this endoneurium is going to wrap itself around the individual neuron, the axon of the neuron, all right? And it's made up of areolar connective tissue, which is a loose connective tissue, all right? And it helps to electrically insulate the axon, which means, all right, the nerve signal that's traveling down the one axon is not going to interfere with the nerve signal traveling down any of the adjacent axons because it's insulated. Right? That can become a real problem, especially if you're dealing with a specialized nerve. Right? That could be a real problem if you start triggering, right, random neurons with action potentials. Okay? So it electrically insulates that axon and it helps to separate those axons from the other axons. Okay? So now what we do is we take a group of axons and we bundle them together. And that's a fascicle. Okay? Could be 10, could be 20, 30, 40, 100, whatever. Okay? So we take a bunch of them, we bundle them together, and then we wrap that up. But this time we're going to wrap it up in the dense irregular connective tissue. Okay? That dense irregular connective tissue, all right, helps to protect it, helps to give it a little bit of support and structure. Okay? And that's called the perineurium. Now near the perineurium you'll have blood vessels. So we're starting to see some blood vessels, all right, to help give nutrients, all right, to our tissue. And then we're going to take all those fascicles and we're going to wrap those up all together. And we're going to wrap them up into one entire nerve. Okay? And the same thing, we're going to coat that with a dense irregular connective tissue. Again, to help to protect it, to help provide it with support and structure. And that's the epineurium. That's the outer layer there. Okay? So again, we will find blood vessels in and underneath that epineurium there. All right, take a quick peek. Here you can see, all right, the layers like we saw in lab. Here's the epineurium, okay? And then inside that epineurium, we can see some blood vessels here in close proximity to the perineurium. And then now we've taken a bundle of neurons, those fascicles, and we've put a covering around that. That's the perineurium. And then we have our individual axon here. All right, and we cover that with the endoneurium, that loose connective tissue there. Okay? All right. Another review slide real quick. When we're going to actually talk about the peripheral nervous system, all right, we're going to talk about the anatomy of the peripheral nervous system, which is made up of nerves, named nerves, yes, but nerves, easy. And I tell folks, try to remember this. If you see a nerve coming off of the brain, okay, the name for the brain region is the cranium. Has anyone ever played that game, Cranium? Yeah. Okay, that's your brain. In fact, I think they still have the brain on the cover of the game. So any nerve that comes off the cranium is a cranial nerve. Okay, that's considered to be part of the peripheral nervous system. Any nerve that comes off the spinal cord is a spinal nerve. That's it. Easy. All right, don't make this more difficult than what it is. Cranial nerve, brain. Spinal nerve, spinal cord. Okay? So the breakdown for the physiology or the functional classification, all right, for the peripheral nervous system is relatively similar to what we saw, all right, when we were talking about the central nervous system. Okay? We've got sensory neurons and we got motor neurons. Therefore, we have sensory nerves and motor nerves. And in some cases, we'll have mixed. Okay? So sensory nerves can only carry sensory information. And we know the direction. Where does the sensory information go? From where to where? Right, it's going to the central nervous system. It's going from the periphery somewhere, so from the receptors, we'll say. So it's going to go from the receptors to the central nervous system. Okay, so it's going to go towards the central nervous system. Motor nerves, all right, can only carry motor information. They're made up of motor neurons. And so they're going to send that information from the central nervous system out to the effector organs, out to the muscles, out to the glands. Okay, good on that? All right. And then some of the nerves in our body, well, actually most of the nerves in our body are mixed nerves. So they'll have both sensory and motor neurons. Okay? So like I said before, when we were talking about named nerves, all right, musculocutaneous, axillary, ulnar, radial, all that, sciatic, those nerves are mixed nerves, which means they've got information going up to the central nervous system and they're carrying information leaving the central nervous system. It's a bidirectional highway. Stuff's coming, stuff's going up and down these nerves, okay? But always keep in mind that a sensory neuron cannot carry motor information. Can't do it, okay? It can only carry sensory information. All right. So we know, and again, just a quick review of ganglion, okay, or ganglia. That's a cluster, all right, of the cell bodies of a neuron in the peripheral nervous system. Okay, that's specific because we call it something different in the central nervous system. In the peripheral, all right, we call it a ganglion. In the central, we call it a nucleus. I think if you remember me telling you that, okay? You thought it was easy when you just remembered that a nucleus was the control center of a cell. That's where the DNA and everything is. Well, somebody got it in their brain to call a cluster of cell bodies in the central nervous system the same thing, okay? And so we know that with a nerve, all right, in the peripheral nervous system, all right, it is a bundle, all right, of parallel axons. Well, in the central nervous system, we call the same thing, a bundle of parallel axons in the central nervous system is called a tract. Okay? Same definition, just different places. Okay. Review of the characteristics. We talked about this last time. Excitability, conductivity, secretion, extreme longevity, and amitotic. Okay? Excitability, the ability of a neuron to react, all right, or respond to a stimulus. Okay, we see that, all right, when we're going to change the cell's membrane potential. The membrane potential, and we'll talk about this probably not this week, I know not this week, next week, is the electrical charge across the membrane. Okay, it has a specific electrical charge. And I'll teach you all about how we establish that electrical charge. Okay, so when we change that, we're going to, normally the cell is at rest. It's not doing anything, okay? But when we change that electrical charge, all right, that membrane potential because of a stimulus that occurred, then we've excited the cell, woke it up. Now it's going to jump into action, okay? Well, that cell needs to transmit that information, that electrical charge, I shouldn't say charge, that electrical signal, all right, from one end to the next end. So it's got to conduct that charge, and that's the next one. Conductivity, okay? It's the cell's ability or the neuron's ability to propagate, that means to keep alive that electrical signal. And it's going to do so through these voltage-gated channels, which we'll talk about. And they'll open up one by one, or they're all next to each other, and they just keep opening up sequentially, okay? When they open up, they let stuff move in and out of the cell, okay? Secretion is going to occur at the end of the neuron. Remember, at the end of the telodendra there's the synaptic knobs. All right, so you got your telodendra, all right, and then you get this big kind of swelling at the end of the telodendra. And inside there, you have all these little bubbles, our synaptic vesicles. And inside those synaptic vesicles, we have our neurotransmitter, okay? Many different types of neurotransmitters, but you have them in there, okay? So what will happen is the electrical charge travels down the telodendra, and then it's going to trigger a response, and these vesicles are going to move towards the end of the synaptic knob, and it's going to release the chemicals, all right, into this gap here. It's called the synaptic cleft, okay? And then you have these gates on the other side. And these gates, all right, which are normally closed, may open up, or it may make it more difficult to open up, okay? We'll get into that later on. So that's going to be the secretion part. The longevity of a neuron, okay, these nerves, pretty much the moment you're born, those nerves that you have are going to be the nerves that you'll have for the rest of your life, okay? They stay with you throughout your whole life, all right? Neurons are what we consider amitotic. They don't divide. Right? Except, there's always exceptions to the rules, and we'll talk about it. All right, there's an area in the brain in the hippocampus, all right, that the neurons can divide. In extreme situations, a neuron can divide, okay? But for the most part, because I always argue with people every semester, they're considered to be amitotic, all right? Your skin cells divide regularly, okay? Neurons do not, okay? So keep in mind, amitotic, they will not divide. All right, we did talk about the cell body before, all right? Just a quick review. Okay, the cytoplasm in the cell body is what we refer to as the perikaryon. That's where we're going to find the nucleus. All right, this is where we're going to start what we call graded potentials. And those are just small, minor electrical charges. For right now, that's not what I'm going to describe it to you as, okay? And these graded potentials are going to try to move towards the axon, okay? So what will happen is, all right, and here's the cell body. You've got your dendrites coming off. And maybe another neuron, its synaptic knob, is inserted onto the dendrite. And it's going to release its chemicals onto the dendrite. And it's going to try to stimulate the dendrite to send information to the cell body. And then that one portion of the cell body is the axon. And it's going to try to send its information to this part of the axon, which is called the axon hillock. And it's going to try to stimulate the axon hillock to generate an action potential, a nerve signal, which will travel down the axon. We'll learn all about that, okay? So in the cell body, we find the nucleus. In addition to that, we find some organelles. Endoplasmic reticulum, okay, site for protein synthesis, all right? On the endoplasmic reticulum, if you see little bumps on there, we refer to that as the rough endoplasmic reticulum. Well, we describe those bumps, all right, as chromatophilic substance or Nissl bodies. Those are ribosomes, right? And as you know, ribosomes, their job is to make proteins. That's what they do. And so they're found on the endoplasmic reticulum, or they can be free-floating. Those are free ribosomes, okay? The dendrites, again, these are short, specifically unmyelinated processes. Myelin is a fatty-like lipid substance, okay? And it's only found on the axon. It's never found on the cell body. It's never found on the dendrites. The only place you see it is on the axon. And it's not even on all of the neurons. It's not on all neurons. It's only on some, okay? Its job is to insulate the neuron. So it speeds up the transmission of a nerve signal. Now, nerve signals travel at milliseconds, okay? It's really fast, by the way, okay? But this myelin speeds it up even quicker. So it doesn't seem like much, but it can speed up an action potential where it would take 5 milliseconds for it to travel from the central nervous system down to your toe. It can speed it up to 3 milliseconds, which is a lot of time in a life or death situation, okay? But that's what myelin does, okay? So dendrites, their job is going to be to receive input, okay, that comes from maybe another neuron or from a receptor. It's going to receive that input, and it's a real simple job, and it's going to transmit that information to the cell body. That's all it has to do. So it receives the input, sends it to the cell body, and it's like this. I'm done. I'm done. That's what I did. My job is done. Now it's the cell body's responsibility to get that input all the way to the axon hillock. Okay? All right. Our axon, this is the longest and largest process that comes off of the cell body. Okay? And so it is going to travel. So here's your cell body, and then here's the axon right here. And it's going to travel all the way to wherever. Okay? And it's going to either connect onto another neuron or an effector organ, which is going to be a muscle or gland. That's it. Okay? Keep it simple. These cells are going to either attach and connect to, I shouldn't say attach, but connect to another neuron or an effector organ, muscle or cell. Okay? So the first part, all right, of the axon is this triangular-like part. I'm going to blow it up a little bit. It's triangular. It's where the cell body starts to taper down. Okay, so here's a larger portion of it. So this triangular part here, that's the axon hillock. All right, that is where your action potential starts. That's a test question. All right, I can guarantee there's 6 people in this room. 4 out of 6 of you will probably see that question on the lecture test. Okay, that is where we initiate. That is where we begin our action potential. And it can never go backwards. It can only go in one direction, and it's down the axon. And I'll tell you why, not today. But it can only go down the axon, never towards the cell body. Okay? So we call the cytoplasm inside the cell body the perikaryon. The cytoplasm in the axon is the axoplasm. And we'll call it something else when we get to the muscle cells. It's called sarcoplasm. Okay? The cell membrane around, all right, the axon, all right, the plasma membrane, is called the axolemma. Axolemma. Now, some of these axons will have what we call collaterals. They're these little branches that come off. Okay, sympathetic neurons have lots of those. So a signal can be coming down the axon, and it can continue to go down the axon, but and then it can branch off too, and go into different directions. So one neuron can affect several different structures. Okay, and these collaterals allow that to happen. So at the very end, all right, of our axon, all right, we got like these little tethers that are coming off. All right, that's the terminal portion, and at the end of those tethers are those swellings there. Okay, well, these tethers are the telodendra. Okay, and those swellings at the end, those are the synaptic knobs. That has the synaptic vesicles. The synaptic vesicles have the neurotransmitter. Okay, so if you want to boil down, all right, the function or the responsibility of an axon, this last bullet point tells you exactly what it does. Okay. Its job is to conduct the action potential from this, well, not from the cell body, but it's from the cell body end, all right, all the way down to the synaptic knob. And then from there, all right, the synaptic job is to release, which is still part of the axon, to release the neurotransmitter. Okay, that's it. Boil it down and make it that simple.
The branches, the axon collaterals, is
Yep.
that like the dendrites?
No, that is not. Just think of it as like another axon. Dendrites are short. Axon collaterals can be quite long. Okay? It's like a road. You know, where the road splits off. Okay, now you come to the fork in the road.
Do they connect other?
It can. It can go to another neuron. Yep. It can go to any of these structures here. Yep. All right. Let's talk about something new. We haven't talked about synapses. Okay, we've talked about all the other stuff. Well, let me just go back to this one here. All right. All right. So you can see here, here's our cell body, okay, with the dendrites coming off, and then the axon over here. But we've got another neuron here, right, that's actually contacting it. Okay, you see the telodendra here, and then the synaptic knobs, okay, that are attaching to the dendrites and the cell body. Okay. Then we have our axon, and you can see here's an example of an axon collateral. Now this structure right here, that's a neurolemmocyte or a Schwann cell. That's only in the peripheral nervous system. That's the one that makes the myelin. So it insulates, all right, the axon, so we can transmit the signal faster. Okay, it's only found in the axon. Then at the other end, we can see, right here, it tapers down into our telodendra, and then again, in this instance, this neuron is synapsing or contacting another neuron. So speaking of synapses, you can see an example of one right here. So you got your synaptic knob, all right, in very close proximity. Now, at this view, it looks like it's actually touching the cell body or touching the dendrite here. But what we're seeing is that the synaptic knob kind of hovers right above the actual neuron okay, the dendrite of the cell body. So there's a gap here, and that's a synapse. All right, a synapse is where you get not contact, but you get one neuron that is going to come in close proximity to a second neuron. All right, so you're going to see in the synapse, all right, what I told you, the purpose, not the purpose, but the axon, all right, is either going to connect to another neuron or an effector. And that's what we see here. So that connection there, all right, is called the synapse. And there's two types that we're going to deal with. Chemical, which we're going to spend most of our time on, all right, and electrical. So we're going to mention electrical here, but then we won't talk about it too much more after this. All right. Chemical are going to be the one that involves the neurotransmitter. All right, they're far more common in the actual body, so we need to have a good understanding. Electrical, all right, is pretty much going to be found just in the brain and the eyes. All right, and whenever you have a synapse involved, you're going to slow down the nerve signal. All right, the timing of the nerve signal just a little bit. All right, but with electrical, you don't. There's no delay, okay? No synaptic delay. Because it takes time for us to transform an electrical signal into a chemical one and then back into an electrical signal. And that's what happens at a chemical synapse, okay? So these two terms here, presynaptic and postsynaptic neurons, all right? Simple. A presynaptic neuron is the first neuron, okay? The postsynaptic neuron is the second neuron. So what that means is, right here, this is what we're looking at. Here's your pre and postsynaptic neuron, okay? This is the presynaptic neuron, okay, because it's before the synapse. Here's the picture of your synapse, and this right here is the postsynaptic neuron. This is the second neuron, okay? So basically, when we're talking about an electrical synapse, we're not dealing with the synaptic vesicles, all right? So you'll see electrical synapses, all right, in the brain and in the eyes, specifically in the eyes because of the speed of that phototransduction, that visual information that we're getting, all right? So there's absolutely no delay, and it deals with this structure here, gap junctions, which we'll see again more in cardiac physiology in 211, okay? So a gap junction is essentially 6 proteins, all right, that are in a circular configuration with a hole in the middle. This is an awful drawing, all right? So you can see we have a hole right here in the middle. And these gap junctions, all right, are located in the cell walls, okay? And there's a hole in that cell wall so that electrical information can pass right through it. All right, chemical synapse, okay? This is the one that's much more common, that we see much more frequently. All right, so we're going to, again, when we're talking about a synapse, we're always talking about 2 neurons, a presynaptic neuron and a postsynaptic neuron, okay? The presynaptic neuron is going to transmit information to the second neuron, the postsynaptic neuron. That postsynaptic neuron receives that information, okay? And it can be, that synapse can be on the dendrites, it can be on the cell body, okay? Well, anyways, I'll try to draw it. All right. Here's our synaptic knob, all right, from one neuron, and then here is the dendrites, or the surface of a dendrite from the second neuron. Anyway, I almost had it. There's a space in between both of those neurons. That is called the synaptic cleft, okay? That is where, okay, the chemicals that are inside the synaptic knob are going to be secreted into. They're going to secrete the neurotransmitter into this space here, this cleft, okay? It's filled with fluid, okay, extracellular fluid, all right? So let me tell you a story as you're writing things down, okay? The events of the synaptic communication. Okay? So it's a quick story. You've heard me tell it once or twice, okay? But repetition is very helpful. All right? So as our nerve signal comes down the axon, it's going to stimulate some things to occur that we'll talk about later. But all in all, it's going to stimulate the secretory vesicles that are floating around, just hanging out, minding their own business. They got their neurotransmitter there. It's going to stimulate those synaptic vesicles to move towards the edge here of the synaptic knob. Once they do that, they fuse with the plasma membrane there, okay? And when they fuse with the plasma membrane, they release their contents out into that fluid-filled synaptic cleft. And whatever that neurotransmitter is, right now we'll just call it chemicals, all right? Those chemicals will diffuse across the synaptic cleft, okay, to a gate of some sort. It has to be a chemically gated channel, okay? It'll move across, and if it's the right chemical, and that gate has the right receptor for that chemical, it'll stimulate that gate to open up. And when that gate opens up, all right, some sort of substance can now move across that gate and into the second cell, into the second neuron, okay? Well, the time it takes for all of this to happen, all right, is what we refer to as the synaptic delay. It takes a little bit of time, okay, for this to occur. Okay, that's why a chemical synapse, there's a slight delay, slows down the signal. Electrical, we don't worry about that. Okay? So when these chemicals flood into the second neuron, they'll start to create what we call a graded potential. All right? And there's different types of graded potentials. One can stimulate the neuron or one can inhibit the neuron. And we'll get into that later on. Okay? So that's what we're talking about here, the postsynaptic potential, the graded potential. So here's what a synapse is. All right? What is the mode of transmission in a chemical synapse? All right? You have a molecule that's stored in the synaptic vesicles that are going to be stimulated from the action potential to move towards the plasma membrane of that synaptic knob, fuse with the synaptic knob, release the chemicals or the neurotransmitter into the synaptic cleft. The neurotransmitter will migrate or diffuse across the synaptic cleft to the second neuron, to the postsynaptic neuron, to the chemically gated channels there, and it'll stimulate those chemically gated channels to hopefully open up and let in some sort of substance that's outside the cell. And that's what happens at a chemical synapse there, right? We'll get into that in a little bit more detail. I'm sure you're excited. So now it's review time, okay? Let's review pumps. We're going to review pumps. We're going to review channels, because we talked about them before. Remember we talked about two different types of pumps before in chapter 4? We talked about the calcium channel pump that pumps calcium out of the red blood cell, so the red blood cell's membrane doesn't harden, okay? So it's flexible when it has to squeeze in through places. Well, the other pump that we're going to talk about is the sodium-potassium ion pump. Think of it as a bilge pump, okay? This pump is constantly pumping sodium out of the cell, out of the neuron. And it's constantly pumping potassium into the neuron. That's what it does, okay? And since it's a pump, it requires energy. And this pump specifically requires 2/3 or 66% of a neuron's energy expenditure. That's a lot, okay? But you're going to find out why, okay? This is what's going to happen. And so, and if you guys remember from chapter 4, I said when this pump is when it's working, all right, when it's pumping sodium out of the cell and potassium into the cell, it's pumping these elements, all right, these electrolytes against their concentration gradient. Because remember, outside the cell, there's more sodium, Okay, and what naturally wants to happen, all right, with diffusion. Okay, you want to go from an area of high concentration to an area of low concentration. So sodium wants to enter the cell, and there's more potassium inside the cell than outside the cell. So potassium wants to leave the cell. But what this pump does is it pumps those substances against their concentration gradient. It's going to keep pumping sodium out of the cell, even though it wants to go in the cell. It's going to keep pumping potassium out of the cell, into the cell, even though it wants to leave the cell. Okay? So you need to know this. Pumps require energy, and they're going to move substances against their concentration gradient. Okay? So we're going to focus on mainly the sodium-potassium pump. Well, and we will talk about the calcium pumps. I forgot. Yeah. Calcium pumps come into play, not just with the red blood cells, but down at the synaptic knob. Foolish me. I forgot. All right. So, pumps. Feel good about that? Okay? Because we're going to talk more about it in a little bit. But now, the other type of transport protein that we're going to talk about are channels. Okay? And there's 3 types. We're going to get into that. Right? But think of channels as a hole in the plasma membrane. Okay? We describe them as a pore, but a hole is fine. Okay? So it's this hole that we put into the plasma membrane, and it's going to allow, all right, ions, which we all know are charged particles, charged elements, all right, charged particles, an electron and a proton, okay? But it's going to allow these ions specifically to move down their concentration gradient. Well, if you're moving down your concentration gradient, that's a passive form of transport. Okay, an active form of transport requires energy. Okay, so pumps require energy. Okay, but passive transport mechanisms, right, when things are moving down their concentration gradient, which means we're going from an area of high concentration to low concentration. Right, so like I said, sodium, there's a lot of it outside the cell, and there's not much of it inside the cell. So what we do is we start poking holes into the cell membrane, and now sodium can go down its concentration gradient and flow into the cell. Okay? So these channels, and you need to know this, are specific, all right, to one type of ion. Okay? A potassium ion cannot travel through a sodium channel. Okay? A calcium ion cannot travel through a potassium channel. So each channel is specific to one type of ion, and that's only when they're open. When it's closed, nothing's getting through. Okay? When it's open, only the ion that it's specific for can get through. So let's talk about the 3 types. The first one is easy to remember, leak channels. Okay? They're always open, like 7-Eleven, 24/7, open for business. Stuff's coming in or stuff's coming out. All right? So we see leak channels, all right, for sodium and potassium. So you're going to have constant diffusion through these leak channels as long as there's a concentration gradient. Okay? So as long as there's more of whatever on one side of that channel than on the other side, you'll see things moving. But if there's equilibrium, if it's equal amounts, all right, inside the cell and outside the cell, will we have diffusion? No, of course not. Okay? If we reach equilibrium, there's no diffusion because you have equal amounts. Okay? There's no concentration gradient. Leak channel's easy. Okay? Chemically, pay attention to this. Chemically gated channels. The first name here tells you that you need some sort of chemical to affect this channel. Okay? So if we see voltage gated channels, then we need a change in the charge, all right? Something that has to do with an electrical charge is going to affect this channel. Okay? So in your minds right now, if you're dealing with a chemically gated channel or voltage gated channel, consider those channels to be closed. Okay? They're closed. So when they're closed, nothing's moving across. So when we talk about a chemically gated channel, we need a chemical to open up that channel. And when we do, then that channel opens up and an ion or some sort of substance can move across the channel. Okay? Is leak channel ion specific? Yes, yes, yes. And you're going to find out these other channels are too. Okay? They're going to be, you're going to have diffusion is going to affect the leak channels. Diffusion is going to affect the chemically gated channels, and diffusion is going to affect the voltage gated channels. We just have to figure out a way to open up these channels. Leak channels are always open, so you don't have to worry about that. Okay? But chemically gated channels and voltage gated channels are usually closed. Okay? So in order for us to open them up, when we're talking about a chemically gated channel, we need a neurotransmitter. Okay? Some sort of chemical. That neurotransmitter will open up that gate, and then that ion can now diffuse down its concentration gradient. Okay? For voltage-gated channels, all right, we're going to need a change in the charge, the electrical charge, all right, of the plasma membrane. And when that occurs, same situation. That'll open up the gate, and then an ion now can now diffuse across it can diffuse down its concentration gradient. So the diffusion applies to all of these channels. Okay? All right, give me a second to take a quick sip. All right, so let's talk about the first type, all right, of channel. It's a specific channel, okay? It's the the voltage-gated sodium channel. So let's dissect it real fast. If it's voltage-gated, that means we need a change in the charge, okay? And now, this is nice because it tells us what is going to move through the channel. So if we're talking about a voltage-gated sodium channel, what that means is we need a change in the electrical charge, and the only thing that can move through this channel is sodium. So if we have a voltage-gated potassium channel, then only potassium can move through that channel, okay? All right. So in this situation, well, when I say this situation, when we're dealing with this type of channel, it's a special kind of channel because this channel has two gates, okay? The other channels I was talking about, all right, most of those except for this one have one gate, okay? But this one is special. It's got two gates. So I'm going to go back and forth through these three states here, all right, from this slide to the next slide, okay? So the first state is the resting state. So when you're sleeping at night and your leg muscles aren't moving around unless you have restless leg syndrome, okay, the nerves, all right, the neurons that affect those skeletal muscles are at rest. They're not doing anything, okay? So we're not having any change in the electrical charge. They're resting, okay? What that means is, all right, the voltage-gated sodium channel, all right, that the activation gate is closed. Now I'm going to confuse you here for a second, so I got to jump over to this slide here, just so you're not confused. Remember I said there's two gates, right? Let me show you the two gates, and then I can go back. All right. So here's one gate, this little swing arm thing. Okay, that's called the activation gate. The other gate are these two little swellings on each side. Okay, so here's your sodium gate here, or channel here. All right, and you have these little swellings on the inside or near the inside of the cell. Okay, all right. So when there's a space between them, like you see here in this picture, there's a space here. Okay, it's open. That's the inactivation gate. When that gate closes, right, those swellings just enlarge, and they enlarge so much that they block up the channel there, and nothing can get down through, okay? So you have an activation gate and an inactivation gate. Those are the two gates. You guys cool with that? Okay? All right, so let's go back to the resting state. So in the resting state, when you're not doing anything, when that neuron is not doing a darn thing, The activation gate is closed. That little swing arm has swung up and closed off the gate. Nothing can get down through. The inactivation gates, those swellings there, all right, they're open. They're relaxed. They're not doing anything, okay? But we're not seeing any sodium enter into the cell. That's what we see here in this picture, okay? This is the outside of the cell, okay? The interstitial fluid, this is the inside of the cell. The cytosol, all right? You see you have all this sodium outside and just this one little guy on the inside. Sodium wants to go down its concentration gradient. It wants to go inside the cell, but it can't because this gate is closed, okay? That's the resting state, okay? The gate is closed, nothing's happening. So now what happens, okay? The second state, all right, the activation state, something causes that activation gate to open up. Well, we know what that something is. Something having to do with an electrical charge, okay? So we get a change in the electrical charge across the plasma membrane, okay? A voltage change. And now the activation gate opens up. The inactivation gate is still open. So now sodium can move in, okay? Right here. So something changed the charge, okay? Well, I didn't point this out on the first slide here. If you look back to the resting stage here, you can see you got a plus sign on the outside of the cell and a negative sign on the inside of the cell, okay? That has to do with what we call the membrane potential. So normally at rest, the outside of the cell is more positive and the inside of the cell is negative, okay? So for our purposes right now, what we've done is we've flipped it, okay? Now the inside of the cell is more positive and the outside of the cell is more negative. That's that voltage change that I was talking about. That voltage change caused the activation gate to open up, and now sodium enters into the cell, okay? And it diffuses down its concentration gradient, okay? So what happens now is once we reach equilibrium, like we're seeing here, then the inactivation gate, those two swellings, they swell up and they close off the gate, okay? So when we get to the third stage, the inactivation state, the activation gate, that swing arm gate, it's still open. It hasn't closed yet. But the inactivation gate, little swellings there, all right, they swell up and they close off the gate. So now we can no longer get sodium to enter into the cell. And we'll talk about what causes that, okay? But this is very brief. I mean, I'm telling you, the inactivation gate being closed is like a blip, quick, okay? As that happens, all right, we're going to reset everything, okay? And we're going to return back to the resting state. Okay, so this closes for a split second, no more sodium can enter, and then the activation gate is going to swing closed, and the inactivation gate is going to relax, all right, and those swellings will go away. But we're still not going to have any entrance, all right, of sodium, okay, because one of the two gates are closed. Anytime you have a gate closed, nothing's coming in and nothing's leaving, okay? Both gates have to be open for anything to travel through. And keep in mind, it's only going to travel through when there is a concentration gradient, okay? If you have more in one area and less in another, that's the only time, okay? All right. All right, modality-gated channels. All right, again, these gates normally close, but they are going to open from some sort of stimulus. All right, and that stimulus is either going to be a chemical, right, a neurotransmitter, or a voltage change, like we just saw there. All right, when we flip the charge of the plasma membrane. Okay. So we're going to see these modality-gated channels, all right, not mainly, but in our sensory neurons, especially those that are going to be monitoring the internal and external environments, all right? So a perfect example is going to be the not the Pacinian corpuscle, although that can apply, but the Merkel's receptor, all right? Well, when you press on that from light touch, it's going to release, all right, a neurotransmitter, all right, that is going to stimulate, all right, that receptor in response to that mechanical pressure that occurs, and that neurotransmitter is going to stimulate the openings of those chemical gated channels. When we get enough of them to open up, then it's going to signal the nerve and create an action potential, okay? But we'll talk about that. All right, so now you've heard me talk about the different types of channels, okay? We have leak channels, and that applies to both sodium, potassium, all right, ions. All right, those channels are always open, okay? We'll have chemically gated channels, voltage gated channels, and our pumps. So now we're going to learn where on the neuron are we going to find these different transport protein mechanisms, okay? So in the entirety, the whole neuron, okay, you will find leak channels everywhere. Cell body, okay, dendrites, axon, okay? You're going to find leak channels throughout the entirety of the neuron. Something that you want to commit to memory right now is you have far more potassium leak channels than sodium leak channels. So we're seeing more sodium diffuse across the plasma membrane. Remember, there's more sodium inside the cell than outside. So you'll see through these leak channels lots of sodium. Did I say sodium? Excuse me. Lots of potassium, all right, leaving the inside of the cell to go outside the cell. All right? So meanwhile, while these leak channels are letting sodium and potassium just leave the cell or enter the cell at the same time, all right, the sodium potassium ion pumps are like, well, that's messed up. That's like being in a rowboat, all right? Two people are in a rowboat. One person's operating a bilge pump, and they're pumping or bailing water out, and the other person keeps putting holes in the boat and keeps letting the water go in. All right, these leak channels are going to let, for example, sodium leak channels are going to let sodium, all right, enter the cell because there's more sodium outside the cell than there is inside the cell. Well, the sodium potassium ion pump's like, the heck with that. You know, I got to keep pumping this sodium that you're leaking into the cell. I got to keep pumping it out of the cell, okay, because that's what I do, okay? So there's a reason why that's going on, and we'll get into that. But just understand, all this stuff is going on all the time. All right? Sodium leaks in, potassium leaks out, and then meanwhile, the sodium-potassium ion pump is bailing sodium out of the cell, and at the same time, bailing potassium into the cell. Okay? So this whole thing helps to maintain what we call the resting membrane potential, RMP. All right, I know I keep saying we'll get into this, but I don't want to throw too much stuff at you. Okay? So for right now, you're going to see these channels throughout the entire neuron. Okay. Good so far? All right, we won't be going too much longer. All right. So I want to kind of talk about the functional segments of a neuron. Okay, you've heard me talk about the structural segments, the dendrites, the cell body, the axon, the teledendra, all right? But we're going to talk about the functional segments, okay? Like what is going on in the cell body? What is going on in the axon? What is going on in the synaptic knobs, okay? So the receptive segment, and you probably already can figure this out on your own, the receptive segment, that's going to be the part of the neuron that's going to receive information. Input is going to come to that area. So that's going to specify the dendrite and the cell body. So both of those structures are going to make up the receptive segment. So those structures are going to receive input, okay? The initial segment is the axon hillock. Okay, the axon hillock is where we're going to initiate the action potential. Once that's initiated, it's going to be propagated down the axon. It's going to travel down the axon, all right? Like an electrical charge moves through the walls here on the wires in the wall, okay? So the axon, all right, and the teledendra are referred to as the conductive segment. They're going to transmit that action potential in one direction only, away from the cell body, down to the synaptic knobs. Okay, so that includes the axon, any of the axon collaterals, okay, and the teledendra. And then finally, the last part is the transmissive segment, and that's the synaptic knob. Transmissive segment is going to transmit that action potential signal to another structure, to another neuron, to a muscle, to a gland, okay? And that's what we see the synaptic knobs doing. So you want to know these terms here: receptive segment, initial segment, conductive segment, and transmissive segment. So the channels, all right, and the pumps that we're going to see, all right, in these segments varies now, okay? So when we're in the receptive segment, okay, we are only going to see chemically gated channels in the receptive segment, which means the cell body and the dendrites are only going to have chemically gated channels, okay? They're not going to have calcium ion pumps. They're not going to have voltage gated calcium channels, okay? You will only find chemically gated channels here. Okay, an example is the sodium and the potassium, the chemically gated sodium and the chemically gated potassium channels. All right, you'll also see a new guy, chloride, okay, chemically gated chloride channels. Okay, in the initial segment, the axon hillock, this is where you're going to see your voltage gated sodium ion channels and the voltage gated potassium ion channels, okay? Conductive segment, again, we'll see voltage-gated sodium channels and voltage-gated potassium channels. And then finally, when we get down into the transmissive segment, down by the synaptic knobs, that's where we're going to see our potassium, I can't talk tonight. That's where we're going to see our calcium ion pumps. Hey, Doc, how you doing?
Good.
Good to see ya.
Don't you love the cone of shame?
I'm going to wear mine.
Now, these things go to Ollie's. You get one that's a lot more comfortable.
Really?
$3.
Oh, I don't know how to do that. Come here.
I'm afraid to.
Left. You got to see. That's the first teacher that I ever taught with here. Beth Moorbach. Awesome teacher. All right, sorry. Okay, so the last portion here is going to be the transmissive segment. That's where you're going to see your voltage-gated calcium channels and voltage-gated, and the chemically gated calcium pumps. Golly, I almost need a break now. She threw me off my game. It's all good. So this figure or picture is in your chapter. I would strongly urge you to take a peek at this, okay? On the left side here, you can see all the pumps and channels, all right, for the entire neuron. Sodium, potassium ion pumps, sodium leak channels, potassium leak channels. That's going to be found in the dendrites, in the cell body, in the axon, in the transmissive segment. And then on the right side, it breaks it down for each region, okay? Receptive segment has these, all right? The initial segment has these, okay? It's just a nice picture to kind of take a peek at. Okay, you can see all the way down here at the transmissive segment, voltage-gated calcium channels and calcium pumps. Okay. All right. So what is the difference between a chemically gated channel and a voltage-gated channel in terms of how they function? In order to get a chemically gated channel to operate, we need a chemical. And in our case, a neurotransmitter. You need a chemical of some sort. In a voltage-gated channel, we need a voltage change to get it to open up. Simple. Okay. All right, good so far? Really want to, yes, I'm gonna start on this, but I won't talk very long. I want you to remember this, and I meant to tell everybody at home this. Banana floating in the ocean. Think about it. I want you to close your eyes for a moment. I'm going to close mine. I want you to imagine a banana floating in the ocean. Okay, do you see it? Do you see it? Okay. What do you think about with a banana? Other than it's yellow and it's potassium. What do you think about the ocean? Salt water. Okay? Sodium chloride. Okay? So the banana is floating in the ocean. Okay? Think of that as your cell. Okay? So inside the banana is where the potassium is. Outside the banana in the ocean is where you're going to find your sodium and where you're going to find your chloride. Savvy? You good? Cool. As long as you can remember that. All right? This next slide will help to make some sense. Okay? So in order to start to understand, all right, about this term right here, membrane potentials, what that is, we have to understand what's involved in creating a membrane potential. And that involves substances. Okay? That involves sodium. That involves potassium, chloride, calcium. Okay? But in this case, we're only going to function, all right, on sodium, potassium, and chloride for right now. Okay? So when we understand that, where we find these substances, all right, is essential for how a neuron is going to function properly. Okay? So let's start off with the first part here, the cytosol. What's inside the cell? Okay? You heard me say banana floating in the ocean. The banana is the cell. So we definitely know that inside the cell we see potassium, right? Okay? But I mentioned two other things here. Negatively charged phosphate ions and then negatively charged proteins. Okay? That's important because when we start to talk about the membrane potential of a neuron, all right, we're going to learn that on the outside of the cell, an arresting neuron, the outside of the cell is going to have a positive charge to it, an arresting neuron. On the inside of the cell, it's going to have a negative charge, which means there's more negative stuff on the inside of the cell, all right, and there's more positive stuff on the outside of the cell. That's why it's more positive outside, more negative inside. So that leads me to go back to here. Negatively charged phosphate ions and negatively charged proteins. Both of these structures are, one, negatively charged, and two, big. So big that they can't leave the cell. Okay, so they're stuck there. And guess what? They're stuck there with their negative charge. They can't leave, so they and their negative charge stays inside the cell. But the only thing that can leave is potassium. Okay, and potassium is positively charged. Okay, so if potassium is positively charged and it starts to leave the inside of the cell, will that make the inside of the cell more negative or more positive? Let me repeat myself. These guys, all right, the phosphate ions can't leave, the proteins can't leave, and they're negatively charged. Okay? So you got a positively charged potassium inside the cell, and it's going to leave. So when it leaves and there's less positively charged potassium inside that cell, does that make the inside of the cell more negative or more positive? More negative. Right on. Yep, that's right. Because remember, potassium leaves, it's taking its positive charge with it. So it'll make the inside of the cell more negative. Okay? So now let's go to the outside of the cell. All right? Outside of the cell, which is the interstitial fluid. Okay? You've heard me say this tons of times. Sodium, all right, is more prevalent outside the cell. Okay? It's positively charged. Chloride, all right, is negatively charged, but there's not too much of that outside the cell. There's still more of it outside the cell than inside the cell, but it's not as much. There's tons, and when I say tons, there's tons of sodium outside of the cell in the interstitial fluid. So that's why banana floating in the ocean. Okay, so sodium is going to predominantly give the outside of the cell most of its positive charge. Okay, and then these two guys here, all right, the negatively charged phosphate ions, the negatively charged proteins are going to give the inside of the cell predominantly its negative charge. All right, That's a lot to chew on, but we're going to stop here. Not too bad for the first day, okay? Believe it or not, it will get a little bit easier, okay? But let's take a break, okay? And for you folks